Assalamu alaikum. Hello guys, welcome back to my channel. Today I will explain about some extra organelles. I will complete the organelles that I explained about. Let's do a simple quick review. As we said at our past lecture or past video that there are sorry that there are two types of organelles, membranous and non-membranous, and we spoke about the nucleus, the general organelles of cell, the cell membrane, the composition of cell membrane, the structure of cell, the shapes. As we said, the nucleus may be elongated or kidney-shaped nucleus, or it's most located in the nucleus. But in some cells, such as epithelial cells in a pap smear, the location of nucleus may be different from other cells. So we explained a lot of things. One of those that were what's nucleus and chromatin. We say that there are two types of chromatin, hetero and euchromatin. Simply, we say that we spoke about the difference between rough ER and smooth ER. If you want to give a review, you can get back to the video and watch it again. So, centrosome and centrioles. They are located near the nucleus, but in some epithelial, the centrioles are located in the apical cytoplasm beneath the ciliated surface. So centrioles are called basal bodies. So basal means base. Nearly, they are at the upper part of this, near the membrane. They are located near the membrane. So those are called basal bodies. This is then an image of centriole under microscope, electron microscope. It's easy. It's just like a cylindrical shape. It's recognized very easily from the other organelles. Okay, so the function of centrioles: formation of formation of mitotic spindle. Mitotic spindle means we know that. We know that it has an important role on mitosis or cell division. Microtubular organization center, cytogenesis formation. So how cytogenesis is formed from the procentrioles and from the procentrioles formed from the procentriol organizer. So cytoskeleton are simply bone of cell. Simply a definition, they are just like a bone of cell, which are composed of complex network of minute filaments. Its function is it maintains cell shape and stability, and they're responsible for some cell junction functions. It includes cytofilaments and microtubules. So, cytoskeleton maintains the cell and stables how we have bone, for example, and it has a role, of course, at cell movement too. Cytofilaments are thread-like minute structure which are divided into three types. We have three types of cytofilaments. So in every cell we have filaments: actin or thin filaments, intermediate, and myosin or thick filaments. Okay, as actin or thin filaments, it's found in muscle cell core of each microvilli, in motile cells such as, for example, nerve cells and in the macrophages, and its diameter is about 5 nanometer thickness in diameter thickness. So here is the actin. Sorry, I say actin. I'm getting confused sometimes. It's actin. Yeah, it's not actin. I say actin. I mix between them, those two words. It's actin. Okay, this is the nucleus, and this is the actin filament. It's written here. So intermediate, they are 10 nanometer thick in diameter. So add 5 to each filament. They include neurofilaments in the neurons. They are called neurofilaments in the neurons, and glial in the astrocytes, and tonofilaments in epithelial cells. So here is the keratin intermediate filament network. This is the nucleus, and those are the filaments. Let me say something, an important note that I didn't write it here, but I will tell you that intermediate and thin filament or actin come together at the same cell mostly. Okay. Microtubules. Sorry, myosin. Myosin, they are 15 nanometer in diameter and occurs mainly in muscle. Microtubules are hollow tubular structure of variable length. So the length isn't specific, there are different lengths, and constant diameter of 25 nanometer. So microtubules are stable permanent structure in cilia, flagella, centrioles, and basal bodies. We have microtubules in them. Okay, so each microtubule is made up of a protein called tubulin. We have alpha and beta, so it's called a protein tubulin that organizes into protofilaments. A total of 13 protofilaments comprise the wall of microtubule. Comprise means it makes or produces, we can say, the wall of microtubule. Okay. Protofilaments, this is important for exam 13, this may come as dashes. The cytoplasmic inclusions, we explained about the organelles at our past lecture. Now, the cytoplasmic inclusions are stored food, pigments, and crystal mostly. Okay, stored foods are glycogens. Glycogens, you know, they are found in. Sorry, I write it oppositely. Glycogens are stored in the hepatocytes of the cell, of the liver cell. So hepatocytes are liver cells. Okay. Chondrocytes are cartilage cell. Okay, you should know those. I will explain about all of them at our future lectures. As you see, here is the nuclei. Okay, those are the hepatocytes. This is an image that shows the hepatocytes. The hepatocytes, as you see, this is the nucleus, this is the cell membrane, and they are attached together. So, as you see, this is an image of the hepatocyte. Simply because I will not explain about them now, but after that we will go to the anatomy of the liver cell. I will explain about all of them. Okay, then the vessels, all in histology. As we see, if we want to see glycogen, we need special stain called PAS or periodic acid Schiff. Okay, periodic acid Schiff. It's written by SH, but we read it as Sh. Okay, SCH. Glycogen, as you see here, this is the glycogen. It's just like magneta and color. I don't know. The color is just like magneta. So as you see, it's written here. It's shown here. I tried to find the slides that are most, clearly seen or clearly visible. Now, glycogens under electron microscope. As you see, B are adipose tissue. B is an adipose cell or adipose tissue. Adipose, as you see, this is the adipose. It's dissolved during H&E, okay? But it is dissolved during H&E. So I wrote it oppositely in my paragraph. No, glycogen is not dissolved. Adipose is dissolved. Okay, adipose is dissolved during H&E stain. So those dark spots are dark dots like structure are called the glycogen. So you see here. So how to differentiate them from ribosomes? They are simply larger. It's very easily, and they are not attached to the rough ER, and they are larger in size. Those are the adipocytes B, and the dark dots are the glycogens. See, so yeah, we will come to the lipids or adipocyte cells, how they appear. This is an example of some histological slides in different animals. They are simply the same, but the size is different. Okay, the thing that adipocyte is dissolved, as you see, there isn't any nuclei or any cytoplasm seen. You just see something white. You just see a white surface. Nothing such as nuclei or Golgi apparatus is seen. No, it's just an adipose tissue having molten from or dissolved from H&D stain. Guys, be careful. We will explain at later lectures. I will explain for you lung tissue. Guys, be careful. This looks like lung tissue, but it's not lung tissue. Lung tissue here, lung tissue at those linings are epithelial cells. But you can't see here any epithelial cells because there isn't any nuclei. So they are not cells. They are just adipose, okay? At the membrane, you can see this just like the outer membrane. There aren't any nuclei, but in lung tissue, there's a nuclei, and you can find alveoli and veins, okay? So you should be careful to differentiate between them. So after that, we would come to lipid appearance by using special staining techniques, okay? For lipids, frozen sections are the best for lipids, okay? To are best demonstrated in frozen sections stained by specific lipid methods such as osmium or sodium-free stain. As we see those, this is a specific or special stain for lipids, sodium and osmium. As you see, those dark black are the lipids, and this one are the adipocytes, or you can say lipids. At this is more visible. This is the lipid or adipocyte. Okay, this is how they look like. Okay, let's go to the, as we said, there are in the inclusions there are stored food, and stored food come into two sources such as glycogen and lipids, and there are pigments. The pigments are exogenous and endogenous pigments. The exogenous pigments are they are produced outside the body, such as carotene. Carotene is found in carrot mostly and in vegetables. There's a normal physiological condition. Be careful, not pathological. That means it's normal. It may look, some skin they appear like green. Okay, this is because of the carotene. Okay, this is called carotenemia. What's carotenemia? Are high levels of carotene in blood. It's normal, it's non-pathogenic. Okay, but for example, there are other types of pigment that are endogenous. It means they are produced inside our body, as we see here. They include the endogenous pigments: hemoglobin, hemosiderin, bilirubin, melanin, and lipofuscin. Okay, lipofuscin. Sorry. Hemoglobin is the pigment or iron found in blood that transfers oxygen. So this is simply as I said, the transfers oxygen, the pigment in blood that transfers oxygen, and it's iron. Okay, hemosiderin. Hemoglobin is broken down into hemosiderin and bilirubin. So hemosiderin is another type of pigment. Bilirubin is when the erythrocytes die and they are broken down. Bilirubin is released. Okay, bilirubin is one of the most common causes of jaundice. Okay, you should differentiate jaundice from carotenemia. Okay, jaundice is what's called pathogenic. So, but carotenemia is not pathogenic. Melanin is the pigment that present in our skin and hairs and eye. Okay, lipofuscin is another type of pigment in the cytoplasm, and it's normal, non-pathogenic. It's seen by special staining techniques, and sometimes when a part of cell dies, the lysosome digests it, and lipofuscin is seen after that digestion. Here are some extra slides that I wanted to show you. The pigments lipofuscin, such as neurons and cardiac muscle cells, hence they are referred to as H pigments or wear and tear pigments. As you see, those green-like substances are granules. Green-like granules are called lipofuscin. Okay, so here, as you see in those types of cells, you can see the lipofuscin. Okay, some may ask, what are those lines between the muscle cell? Okay, what are those lines? Those are fibers. Okay, we have some types of fibers that I will explain about: elastic and collagen fibers. I will explain about in other videos. Those are fibers. Those thin like structure, they look like hair. Okay, those are fibers. Okay, this is lipofuscin, as I explained. Hemosiderin. The hemosiderin is a golden brown iron containing. So pigment usually seen within the cytoplasm of the splenic macrophages. Okay, those are the splenic macrophages. The macrophages, are types of white blood cells. This is the hemosiderin. As you see, this spot is the hemosiderin. This, I will try to draw on it. Let me draw on the hemosiderin. This is the hemosiderin. I circled it, as you see here. This is the hemosiderin. I circled it here. The melanin. Melanin is found is brown to black pigment in our hair and skin and eyes. Okay, as you see, those one are that I magnified on are the melanin shown. Okay, those are the melanin, and this is the connective tissue. Those are the fibers seen, but I will explain about them later. So as I told you, this is the melanin pigment. So let's write on it. This is the number one, is the down layer, or you can call that, for example, connective tissue. Those are the epithelium, and this is the melanin that I showed you now. Okay, so thank you guys for watching. Hope you get benefit.